As plastic surgeons, you always know that keloids presents a difficult challenge, particularly because we can't determine those patients preoperatively who are going to develop keloid scarring. And that's one of the reasons why we undertook this study with the aim of determining which genes are implicated in it, with the hope that we can develop prognostic tests and perhaps lay the groundwork for future studies and treatments. So the project objectives are to determine the true inheritance mode of keloid scarring and to identify the genes implicated in this process. Now, the project strategy was a two-pronged attack on this disease. It started off with a clinical characterization and recruitment of patients in Africa, in the Sudan, where we set up dedicated keloid scar clinics. And we also went out to rural regions of the Sudan and collected some more clinical data and biological specimens, which we then transferred across to Manchester, where I did the data analysis and genetic studies. Now, the reason why we chose to collaborate with the University of Khartoum in Sudan is that there's a perceived high incidence of keloid scars in the Sudan, and importantly, the availability of tribes where we can find 4, 5, or 6 generations of one family living in one region that demonstrated keloid scarring. Another important reason why we went to the Sudan was the practice of tribal scarification, and that gave us a parameter by which we could determine true keloid formers from non-keloid formers, as everybody, after a certain age, would have been subjected to a uniform level of dermal injury. Now, we were very fortunate. We were able to recruit more than 200 keloid patients and 3 very large pedigrees with multi-case familial keloids, and we conducted the segregation analysis to determine the inheritance mode. And in 2 of the pedigrees, it was autosomal recessive, and in 1 of the pedigrees, it was dominant. We also conducted a detailed clinical characterization of the disease and uncovered the keloid phenotype-genotype link. We found 2 different phenotypes, a horizontal keloids, this is what we've chosen to name it, which demonstrates irregular superficial subepidermal spread, and that was prevalent in the autosomal dominant pedigrees, and the vertical keloids, which were raised and bulbous, and they were prevalent in the recessive pedigrees. This took us to the gene mapping studies, which we did using whole genome scans with more than 1 million genetic markers. We then did autozygosity mapping, linkage analysis, and homozygosity haplotype analysis. I won't go into details of that because of the time limitations. But to cut a long story short, we're able to find 5 chromosomal regions of interest with susceptibility genes or candidate genes. We further narrowed that down by conducting gene expression studies by taking RNA extracted from keloid tissues and comparing it to RNA from normal tissues. We took 8 keloid affected samples and 4 controls, and 4 of them were from black individuals and 4 from Caucasians. And we found that there was statistically significant difference in the expression levels of 3 genes: KIT, TMEM165, KIAA1211. So the conclusions of this study are there are 2 forms of transmission of keloid disease, dominant and recessive. Clinical characterization reveals 2 phenotypes, horizontal and vertical keloids, which correspond with the transmission mode. Most importantly, keloid candidate genes have been identified: KIT, TMEM165, KIAA1211. Thank you.
Congratulate you to these fascinating and really highly interesting data that you achieved so elegantly, with so much effort going into effort. Now, having these genes, what would be your next step to prove your hypothesis that these 3 genes are really those who might trigger keloids?
Right. We've already started doing that by doing the gene expression studies, which verified that there was a difference in expression between affected and unaffected people from different ethnic backgrounds. Now, the next step would be to do it on a larger number of samples. The third step would be to look at sequencing those genes, and we've already started doing that and finding the mutations that cause it. And that will hopefully help us find the pathway to developing keloid scars, and hopefully we can, in the first instance, come out with a prognostic test, bedside test, before one of your patients goes to surgery. And second of all, maybe perhaps we'll be able to develop drugs that will block this process happening in the future.
Thank you. Good luck.
Thank you.
Dr. Bella?
Just one question, one comment that may be of interest to you. Gypsies, not Egyptians, gypsies, they make no keloid. That is something that may be relevant to investigate. As far as I know, nobody has done it. Gypsies don't make keloids.
Very interesting. Thank you.
Dr. Bella, thank you for your nice presentation. My question is, what is your definition for keloid in this study in terms of just to remove the hypertrophic scars?
Right. Actually, as part of my thesis and part of a publication that was submitted to PRS and accepted, Professor McGraw there and the rest of the team working on this, we've come out with a new definition. I don't know it by heart, but keloid defined it as a persistent raised scar that is recurrent and has certain characteristics, being that it recurs, it does not show signs of regression, it may show mechanosensitivity, it may show pigmentative changes at the peripheries. And in older individuals, it may show areas of central resolution or quiescence. That's about as much as I remember of it from my head, but you'll hopefully see it in the PRS soon. Thank you.
Thank you. Now, the next paper is significant symptomatic relief of scar pain and itch by using a